Hello, my name is Humberto Fortunato. Raquel and I work at Hubs. I'm going to present a curious observation we made during our research on the Golden Mussel project, which aims to evaluate the Limnoperna fortunei infestation at hydroelectric power plants in Brazil. This presentation is entitled Can Freshwater Sponges Naturally Control the Invasive Mussel Limnoperna fortunei in South America? First of all, let me introduce the species we are working with. Limnoperna fortunei, commonly known as golden mussel. This is an invasive species that came to South America from Asia in the beginning of the 90s. It's originally from China and must have arrived here with ballast water in Argentina. Since then, it has been spreading through our freshwater systems, and now it's found in most regions of Brazil, as you can see here in this map, except to the north region. Thought lately, there is some concern that it could be a threat even to the Amazon. Most of the inland dispersal of the golden mussel is aided by boat traffic. This animal lives attached to substrates by the byssus, but the planktonic larvae do contribute to the dispersal downstream. These traits favor the formation of huge aggregations reaching up to 200,000 individuals per square meter, which enhance its dominance in the benthic systems and cause severe ecosystem shifts. The ability to settle on virtually any surface makes golden mussel an undesirable and potentially dangerous organism. Its ability to grow on other organisms has already been evidenced in several works. We can see here some pictures showing the epibiosis of this species carried out on plants, sessile, and mobile organisms, hindering filtration or damaging hard and soft tissues. However, few studies have experimentally shown the effect of this epibiosis on the hosts. Golden mussel also impacts the economy of Brazil. They directly affect Brazilian energy production, reaching almost 40% of the hydroelectric matrix, causing operating expenses in the order of $1,200 million per year. Other important impacts are on fish farming, fishing, tourism, and leisure, as mussels change the landscape of continental beaches. Facing these issues, we started to study the severity of infestation of the species near hydroelectric power plants of Ilha Solteira, Três Irmãos, and Jupiá in the central zone of Brazil. We can see here the sites of these hydroelectric power plants in the map. And this work is to help hydroelectric companies to understand the magnitude of the problem and to seek solutions for the management of these species, since the discharge of chemical products and manual cleaning are ineffective and expensive. To this end, we performed photoquadrats at all sites marked in green on the map, with abundance and biomass counting at each site. And to our surprise, when diving at the sites marked in red, we observed freshwater sponges growing on the mussels, which until now we have never reported. In this new context, I'm going to show you some of the observations we made on the dives performed in December of 2020 and August of 2021. Our goal with this portion of the research is to establish a baseline of observations of spatial competition and perhaps keep track of the progression of this competition or use it as a reference for lab experimentation so we can understand this ecological interaction and how it affects the invasion of the golden mussel. Talking a little bit about the freshwater sponges, they are inside the phylum Porifera, and they can be distributed in both marine and freshwater systems. They are highly variable morphologically. We can see here in this picture that sponges have high tolerance for extreme environments. They produce gemmules to protect a mass of totipotent cells. In Brazil, we have 67 species holding 30% of the entire biodiversity. And some of these species are already in the red list of animals. And ecology is still the main gap in freshwater sponge knowledge in Brazil. To identify freshwater sponges, we use basically external features, but mainly their specular types and sizes. Here you can see 5 different types and sizes of specules from 5 different species. And we can see megascleres as MG, microscleres as MC, and gemuloscleres as GM. Here I will start to show you some of the species we had observed in interaction with golden mussel in the study area. And with the exception of Sterrastrelopsis brasiliensis in the right, all of the images are underwater. And we can see in this first image, Heteromania crystallina growing on the valves of the golden mussel. And we can see here the siphon of the golden mussel. It's a short siphon, and probably when sponges overgrow the golden mussel, they hinders the ability of filtration of the golden mussel. In this sense, they lose the possibility to feed, and consequently they die. Here, other images, other examples of freshwater sponges overgrowing golden mussels, and I would like to show these species of Drulia, a big specimen on the right, and it's growing over several golden mussel specimens. So we found many species of freshwater sponges overgrowing the golden mussel. So what we know about sponges and invasive mussels interaction is basically based on North American studies. The studies on Canada has shown 10% of zebra mussel overgrowing by sponges, and mainly Eonaps fragilis is the sponge that most interact with zebra mussel. 90% of fragilis associated to zebra mussel are able to kill the invasive species by anoxia. Significant reduction in glycogen and wet soft tissue in host mussels was also observed in the US. And in general, freshwater sponges win space competition against zebra mussel. However, zebra mussel is able to reproduce year-round, so they maintain the invasive populations. In Italy, Ephidiatia fluviatis had intensively suppressed zebra mussel growth in both encrusting and massive forms, acting as a biological control agent of the invasive mussel. And here in Brazil, we only had recorded Corvospongilla sect and Tropospongilla happens overgrowing the golden mussel. It's interesting that in our study area, until 2003, there was no golden mussel recorded, and the community was plenty of freshwater sponges. By 2007, these communities were completely replaced by Limnoperna fortunei. So now what we see is as conclusion is that freshwater sponges are probably adapted to the presence of the golden mussel, and they are returning to their past occupancy areas. Our observation highlights the possibility of freshwater sponge to be a natural agent to control Limnoperna fortunei. We found here at least 6 species overgrowing or surrounding the golden muscle valves, which had never been, 4 of them had never been recorded. And we know that we still need an experimental study to corroborate our findings and our hypotheses. And we would like to do this in the future using SAM and lipidomics to analyze the glycogen and phospholipids on these affected golden muscles. This research is part of a larger project on the golden muscle scope that is being developed in Brazil with the support of CTG Brazil, Speak Brazil, Tijuana Energia, and ANEEL, sponsored by the Research and Development Project. And I'd like to thank you for the attention. And if you have any questions, please let me know.